folks, let's try to get started. I have note cards. If you want to come up and get note cards, and please come and get some cake. This is the first time I've ever done that. I've never done this with a class, but I've really enjoyed working with you guys this term. It's been a lot of fun, and I really have had a good time. So I thought it was kind of a fun thing to do. And before I give you all Fs, I thought you might want to have something sweet, you know. So you think I'm kidding? Okay. So seriously, do come and get some cake because I'm not taking that home. My wife will kill me, and that cake would probably kill me as well. So, all right, we have reached the bitter end. We have a fair number of things to cover, and we need to get into them. So, last time I talked about, I got a good start on nitrogen metabolism, and today I'm going to finish nitrogen metabolism. And there's a fair number of things to say in here. So I'm going to go through things at a fairly fast clip, and I know you're probably saying, so what's new? But I'm going to do that, and then we are going to be done with everything. I have scheduled a review session, and the review session will be on Sunday at 5 p.m. in ALS 4001. I'll repeat that. ALS 4001, 5 p.m. on Sunday. Last time was in here. I think we've done one of them in that room. Yeah, I think that's right. Yeah. Okay. The building is supposed to be unlocked. If it's not unlocked, then I will make sure it gets unlocked, but it's supposed to be unlocked, so we should be set. Okay. So ALS 4001, Sunday at 5. I will videotape it. And I will be in my office next week, so if you have questions, come see me. I'd be happy to meet with you. And the exam is scheduled for, I believe, Wednesday at 9:30 in here, right? Everybody's here bright and early thinking very good biochemistry thoughts at that time. Okay. Okay. Well, last time I finished, I was about right here. I just want to say a couple of very brief things about some considerations of some amino acids. One of the amino acids that's very important, I mean they're all important obviously, but one is very important because it's one that requires sulfur, is cysteine, and cysteine is the one that makes the disulfide bonds, so it's a pretty critical amino acid for structural purposes. And cysteine is actually made from serine. Again, I'm not worried about the reactions here. They're very similar in their starting structure, the only difference being that serine has an OH where cysteine has an SH. Okay? So a couple of reactions to get that sulfur onto there. And sulfur metabolism also is something that's kind of interesting with respect to how it goes through the biosphere as well. Okay, so we talked about the nitrogen cycle and so forth. There actually is a sulfur cycle as such, although we don't really think about it in quite the same terms. But sulfur has to be properly handled in the environment by bacteria. And they also are our source of extracting sulfur from the environment. Let's see, where was I? One thing I want to talk about, last time I should have brought it up at the same time I talked about the folates, is I talked about one-carbon metabolism. And one-carbon metabolism is really critical because, again, these are sources of individual carbons that get put onto various molecules during synthesis. Particularly in the case of folates, that's important for nucleotide metabolism and other metabolic pathways as well. This guy right here, S-adenosylmethionine, is actually a byproduct, or is actually used in the synthesis of methionine. And one of the interesting things about it is that there's methionine right there, and you can see S-adenosylmethionine is something that has an adenine with a methionine attached to it, as it is right here. It turns out that the attachment of that methionine to an adenosyl group makes the methyl very good to be donated to something else. So this S-adenosylmethionine is a very, very good source of methyl groups. They're called methyl donors. Okay? methyl donors. And that's the primary source of methyl groups inside of cells. Okay? Let's see. Yeah, I'll leave it at that. Okay. And let's see. Essential amino acids. All right. So nutrition majors, again, know something about essential amino acids. I mentioned last time that they have to be in our diet because we can't synthesize them. What you see are the human requirements for amino acids, the ones on the left being the ones that we require, the ones on the right, ones that we can synthesize ourselves. You'll notice that histidine is one that's essential for children but not necessarily for adults. So we don't absolutely have to have histidine in our diet as adults, but again, that's just sort of parenthetical information. I don't ask you to memorize which is which here, but you should know what it means if I say what is an essential amino acid. When we look at the breakdown of amino acids, the breakdown actually turns out to be at least a little bit simpler in terms of categories. During the synthesis of the amino acids, we saw that they fit into several groups. When we break down the amino acids, we really put them into two primary groups. Okay, so the two primary groups are glucogenic and ketogenic. Okay, so glucogenic are those that make intermediates that can be broken down in glycolysis or can be used to make glucose, because remember we can go either way, glycolysis or gluconeogenesis. And the ketogenic amino acids are those that are involved in making ketone body type molecules and or intermediates in the citric acid cycle. Okay? There are a handful of them that are involved that can go either way. That is, that they are either, they can be, I'm sorry, not either, but they are glucogenic and ketogenic. All right, so they can be metabolized in more than one way to do that. And let's see. The products of metabolism of nitrogen compounds is very important. So I talked about the importance of balancing things inside of our cells, and balance is very, very important. And it turns out that nitrogen balance is critically important. And the reason that nitrogen balance is critically important is because we have this compound here, ammonium, which is the hydrated form of ammonia, those compounds are toxic. So we have to balance ammonia in our body properly. If we take in too much ammonia, then we excrete it in the form of urea. In effect, we excrete it in the form of urea sort of routinely anyway. All right? So we have a sort of inflow, outflow thing. If we start making if we get too much in, then we'll obviously dump more of the amine compounds into urea. Another byproduct of metabolism of nitrogen compounds is very important when we consider it with respect to nucleotide metabolism, and that's uric acid. Okay? Human beings, for example, excrete urea as their primary way of getting rid of excess ammonia and ammonium ions. All right? But other organisms don't do that. So examples include birds. Birds don't excrete urea, they excrete uric acid. And uric acid is kind of an interesting compound because uric acid, we make uric acid in our bodies, but we don't excrete it as uric acid. Okay? It's actually a byproduct of the breakdown of purine nucleotides. That is, nucleotides like adenine and guanine. All right? So as we break those down, we produce this uric acid in our body, and if we don't produce too much of it, everything is okay. That is, uric acid itself gets broken down into other things. But if we produce too much, something really bad happens. It's very painful. And it's a condition known as gout. Gout. Okay? So gout is a disease that results, as I said, from production of too much uric acid. And what happens is uric acid is not very water soluble. That's why birds don't pee. Okay? Birds just basically splat, right? All right? So it's not very water soluble. So if you produce it in your body, what happens is it will frequently come out of solution and it will crystallize. Well, when it forms crystals, it travels to the lowest place in your body which will typically be your big toe. And when it gets to your big toe, those crystals really start doing a number on your nerves in your big toe. People who have gout have very, very painful big toes, okay? Because these crystals of uric acid are accumulating there and absolutely nailing their nerves in their big toe, okay? How many people in here know somebody who has gout? Yeah. So fortunately, in the modern world, there are some medications that you can take that relieve it. In the olden days, before they had the medications, they used to have big comic scenes about somebody holding their big toe because they were just really hurting. And it's apparently a very excruciating pain if you have a bad case of gout. In the old days, it used to be known as the rich man's disease because only the rich were able to afford the diets that gave them too many purine nucleotides. These included red wine, red meat, and in the modern world, of course, everybody eats these things, and so gout is much more widespread than it ever used to be. Okay? Now, one of the interesting things that happens with gout that we've learned in recent years is kind of odd, and we don't know the connection. We don't understand what the connection is. But it turns out that people who have gout appear to have some kind of protection, at least some protection, against multiple sclerosis. So people who have gout tend to have a lower incidence of multiple sclerosis than people who don't have gout. So there's clearly some protective effect that uric acid is giving. And it's thought that perhaps uric acid, which is known to be somewhat of an antioxidant, is actually preventing some sort of oxidative reactions from occurring in the body that arise and give rise ultimately to multiple sclerosis. Okay. And here's what happens to uric acid. If everything's going okay, then you just break it down, ultimately making urea. If you produce things too fast, faster than these reactions can occur, then uric acid precipitates out as a crystal, and that's where you run into the troubles. Okay. The last thing I want to say here about nitrogen metabolism is the urea cycle. So I've got one more cycle to see. And the urea cycle is one that is another circular cycle. It's, along with the citric acid cycle, one of two circular cycles that exist inside of animal cells. And it's there for a couple of purposes. It's there as part of the metabolic pathway that give rise to some of the amino acids. It's also a way for the body to excrete urea if there's too much nitrogen that's there. So one of the byproducts of the cycle is that every time the cycle turns, a molecule of urea is produced. And so the more nitrogen you need to get rid of in your body, the more this cycle is going to run. Now you'll see that this cycle occurs both in the cytoplasm and in the mitochondrion. I will give you some good news. The good news is I'm not going to ask you to memorize which goes to which, goes to which, goes to which. Okay? So you won't have to do that. But I think there are some intermediates there that you should know. Okay? One of which, well, I've already said, is urea. Duh, the urea cycle is going to have urea in it. Another interesting one that pops up here is arginine. Okay? So arginine is actually cleaved to make urea. It's cleaved to make urea. Okay? Now, I'll mention one other compound that's in here, and I mention it because you frequently see it popping up in energy drinks that companies make. And to be honest with you, I don't really know why it's in energy drinks, but it's citrulline. Citrulline is a product of, or is an intermediate in the urea cycle. And I should also point out, sometimes people ask me about, well, are there amino acids that are in the cell that don't make it into proteins? And the answer is there are. All right? Citrulline and ornithine down here are examples. They are both amino acids, but they're not used to make proteins. So they're both used in the urea cycle. So citrulline, ornithine, and arginine and urea. I think those are the highlights here of the urea cycle. Okay? Questions, comments? Okay. Well, in the time we have left, I'm going to talk very briefly about the metabolism of nucleotides. And the nucleotide metabolism, I think, is a fascinating topic because, as I've alluded to earlier in the term, It's important for the cell to balance the relative number of nucleotides properly. If we look at cells that have trouble with that balance, we discover that those cells are much more prone to having mutations. So we know the balance helps prevent mutations. Very important consideration. So as a consequence, cells have very careful controls inside of them to help keep things from getting out of balance. Nucleotide metabolism is probably one of the most, if not the most, regulated pathways that occur inside of cells. All right? Well, if we look at, this is a schematic example of a purine nucleotide. You should know that a purine, of course, has 2 rings. A pyrimidine has 1 ring. We talked about this earlier in the term. You should know that the purines include adenine and guanine. And pyrimidines include cytosine and uracil and thymine. If we look at the purines, we can see that the source of every atom in that purine, where it comes from. Okay? And what we discover is that for the most part, every atom in the purine comes from very simple compounds. Very simple compounds. Okay? In our bodies, this comes from this mouthful of a name, folate, but it doesn't have to. Our bodies happens to use a complicated molecule to make this particular carbon. This is one of the 1-carbon donations I talked about before. But we know if we take the chemicals that existed on the primordial Earth and we mix them together, they will spontaneously make the bases that pop up in the nucleotides. In fact, if we look in meteorites that fall to Earth, we can actually find things like the bases that we have in nucleotides. That tells us that the reactions that make these are very, very simple. It doesn't take a giant orchestration to make the molecules that we find inside of DNA and RNA. Okay? Well, the same thing is true of the pyrimidines. The pyrimidines are very, very simple. I don't have a figure showing you that, but again, the pyrimidines have only a single ring, and the origin of the individual atoms is all very simple compounds that come to make those. I want to just show you something, and by the way, what I'm showing you are little pieces of the overall biosynthetic pathways that lead to the individual nucleotides. I'm sparing you a lot of reactions. There are 10 reactions that lead to purines from the very simplest precursors, and there are about 10 reactions that give rise to the pyrimidines as well. Okay? Well, we're looking here at about steps number 8, 9, and 10 in the synthesis of the purines. Okay? I like to show this part of it because this part of it is the nuts and bolts of what's actually going on with the synthesis of these compounds. Okay? We have an immediate precursor called IMP. Anybody remember where we saw the nucleotide I appearing earlier? I is a nucleotide. It was the wobble base. Okay? That's inosine. All right? So the wobble base actually comes from here. And it's only used in that wobble base. That's the only time it actually is used. But it's an intermediate in the synthesis of both AMP and in the synthesis of GMP. All right? Now, what's interesting is if we look at this, what's happening if we go to the left is we start over here with IMP. We use energy, and the energy we use is GTP again. And we produce AMP. Okay? Over here, we have IMP. It goes over this direction, and we use ATP and produce GMP. This is actually a pretty cool way of balancing things. GMP will inhibit its own synthesis. It'll inhibit this enzyme right here. And AMP will inhibit its own synthesis. This enzyme right here. And no, you don't need to memorize the enzymes. Okay? Now, let's think about this. AMP inhibits its own synthesis and uses energy from GTP to get made. GMP inhibits its own synthesis and uses energy from ATP to get made. This is how we balance the relative amounts of A versus G. Too much G turns off its own synthesis and uses its energy to make A. Too much A turns off its synthesis and uses energy to make G. Okay? Balance is very important, and this balance works really, really well. Now, notice during this synthesis we see there's aspartic acid, there's an amino acid that's playing a role here, and there's fumarate. There is a citric acid cycle intermediate that's actually being produced. Here is an amino acid glutamine being used, and this guy over here actually uses NAD. All right? So we see involvement in the synthesis of nucleotides. We see intermediates from other pathways. And as I've tried to tell you from the very beginning about metabolism, these pathways are not isolated. They're all interconnected. Very, very interconnected. Okay. The overview of this pathway is important. There's a lot of stuff that's there. And you can see all the individual reactions that it's pointing to here that give rise to IMP. It turns out that the very first enzyme in the pathway is an enzyme that is feedback inhibited. And it's feedback inhibited in a very interesting and unusual way. Right? So I'm going to give you the name of this enzyme because it's an important enzyme. All right? The very first enzyme is called PRPP amidotransferase. I'll spell it. PRPP AMIDO TRANSFERASE. PRPP amidotransferase. Catalyzes the very first reaction. Okay? Now, this very first reaction, as I said, this enzyme is feedback inhibited in an unusual way. What does that mean? If I had normal feedback inhibition, the end product would stop the enzyme, right? This guy has two end products. It has an end product of AMP, and it has an end product of GMP. It takes both end products to fully inhibit the enzyme. If only one of them is present, if I only had AMP, or I only had very tiny amounts of GMP but plenty of AMP, the enzyme will only be partially inhibited. Now, at first that might seem kind of odd, but when you think about it, it actually makes a lot of sense, especially as we think about balance. All right? Let's imagine I had a situation where I had a ton of AMP, I had very little GMP, and I need to balance the equation. What do I need to balance the equation? Well, I'm going to need to make a whole bunch of IMP, right? Because IMP is going to go make GMP because this part of the pathway over here is going to be shut off, but I've got to make IMP. If this enzyme were completely turned off, then what would happen is we'd have no IMP. We would never balance the equation. So leaving the enzyme partly active when only one nucleotide is there allows the cell to make enough of this to keep this process over here going and balance the equation. It works really well. Now, this process also shows that the AMP and GMP can go on to ADP, GDP, that means 2 phosphates, ATP, and GTP, that's 3 phosphates. Okay? Now, these all affect a variety of things. We're not going to worry about all the individual inhibitions and so forth. But I will tell you one other enzyme on here that's a really important enzyme. And it's one that's going to be easy for you. Because this enzyme converts every diphosphate to every triphosphate. No exceptions. One enzyme does it all. The one enzyme is known as NDPK. There's a longer name, but I won't give you the longer name. NDPK. NDPK catalyzes the conversion of ADP to ATP. It catalyzes the conversion of GDP to GTP. It catalyzes the conversion of UDP to UTP. You start to get the idea. It also works on the deoxyribonucleotides. It converts DADP to DATP. So every diphosphonucleotide is converted into a triphosphonucleotide with this enzyme. Okay? NDPK, very important in that respect. All right. Catabolism. Breaking these guys down. We've just synthesized them. All right? There's a whole bunch of reactions. We're not going to worry about those reactions because we've already talked about them. There is the most important product that, for our purposes, and that is the synthesis of uric acid. Okay? Uric acid is an intermediate in the breakdown of purines. Too much uric acid, you got gout. The excreted end product in humans, if you're lucky. It's actually excreted more commonly as an end product. And by the way, uric acid is interesting in where it's excreted, okay? So you can excrete uric acid to some extent in your feces, all right, which is what birds are doing, all right? But more commonly, a lot of people don't know this, Dalmatians actually don't excrete urea. They excrete uric acid. Anybody here ever have a Dalmatian? Nobody had a Dalmatian? If you ever have a Dalmatian, or be careful before you get a Dalmatian, because they are very sickly dogs, all right? They usually are in pain a lot of the time. And the reason that they're in pain is they're full of uric acid, because that's what they excrete, okay? And they are a walking case of gout. So people who have Dalmatians will comment frequently how many times they have to take their dog to the vet, because the dog just isn't acting right, just doesn't feel right. The reason is like I say, it's a walking case of gout. Okay. I'll mention something else here briefly. When we talk about nucleotide metabolism, we talk about salvage in addition to synthesis, all right? It's like a salvage yard, right? So we go to a salvage yard because we don't want to pay the price for a new starter if we can buy an old functional starter off of a junked car, right? I used to do that all the time, right? I don't think I ever bought a brand new starter in my life until I actually had a real job and I could afford it, right? So I always went to the salvage yards and picked up starters, all right? Well, the cell does that. The cell can have pieces of nucleotides that it uses to make full nucleotides. This is called salvage synthesis, okay? Salvage synthesis. You can see, for example, what if I had some adenine floating around the cell? When would a cell have this floating around? Well, think about it. We eat other organisms. We take, and the first thing we do is we break down all the things of those other organisms. We break down their proteins. We break down their DNA. We break down their RNA. We break down their nucleotides. We're going to have bases and other things floating around that if we don't use them, we're just going to waste them. So we salvage them, and in salvaging them, we put the pieces together to make things that we can use for other purposes. All right? Now, there's an enzyme that's involved in the salvage of purine nucleotides that is actually this guy right here. You don't even know the name of it. All right? But it's an interesting enzyme because people who have a genetic disease in which they're defective in this enzyme develop something called Lesch-Nyhan syndrome. Anybody know what Lesch-Nyhan syndrome is? It's a disease you don't want to have, okay? This seemingly very simple reaction doesn't work in people who have Lesch-Nyhan syndrome. And if you have Lesch-Nyhan syndrome, you have severe neurological problems. And the most common behavior that people with Lesch-Nyhan syndrome show is that they eat their own lips off. They chew them off. They will chew, they will eat their fingers, they will and it's not completely understood why this happens. They usually have to spend a good deal of their lives restrained, so they're not eating things that they shouldn't eat. Yeah, kind of a gruesome disorder. Okay. I'm sorry. I gave you the wrong enzyme. There's the enzyme, HGPRT. Sorry. That woke everybody up. Okay. So nucleotide metabolism is interesting, guys. Okay? Pyrimidines. Pyrimidines are the nucleotides that contain cytosine, thymine, or uracil. And our cells obviously know how to synthesize them as well. There are all the reactions necessary to make the first pyrimidine nucleotide, which is called UMP. All right? Now, I'm not going to go through the reactions here and explain them to you. I will point out, as always, a couple of things. It takes energy to do this. It takes an enzyme that you've seen before. What does that enzyme do? Well, we studied it earlier when we talked about the allosteric regulation. ATCase was the one that was the first example I gave you of feedback inhibition. Okay? And feedback inhibition with this enzyme was interesting because the end product of the pathway turned this enzyme off. Well, this pathway continues because this ultimately gets made into UTP, which gets made into CTP, and CTP is the end product of the pathway that turns this enzyme off. So what that means is this pathway has a way of shutting itself down. We saw that the other pathway had a way of shutting itself down, that is the purine pathway. If we get too many purines, it's going to shut itself off because that feedback inhibition occurs. If we make too many pyrimidines, it's going to shut itself off too because we can't have too many of those guys. All right? Okay. There's the NDPK I was telling you about before, called nucleotide kinase, but it's actually called nucleotide. It's actually a longer name. It's abbreviated here. But kinases catalyze those conversions. We have a variety of kinases that catalyze this reaction, only one kinase that catalyzes this reaction. Okay? And let's see. There's the conversion of UTP into CTP. So CTP is made directly from UTP. And let's see, what else do I want to say here? So there's ATCase. You see CTP coming back and inhibiting ATCase right there. There are other things that will affect the pathway, but this is the primary enzyme that's regulated, is this guy right here. All right. And breakdown of the pathway of the pyrimidine nucleotides is not nearly as interesting as it is for the purines. Okay, so we don't have things like gout and all and other things that give rise to interesting things there. And I'm not aware of myself of any major problems arising from problems in breakdown of the pyrimidines. Pyrimidines are also simpler. There's only one ring instead of two. They're also simpler than purines, and so it may be a reflection of that as well. Well, the last thing, there's the magic word you've been looking for all term. The last thing I'm going to talk about this term are the deoxyribonucleotides. Everyone want to stretch first? You want to take one last stretch before? No? Get it done. All right, all right. We'll get it done. All right. Let's talk about how do we make deoxyribonucleotides, all right? Well, three of them are easy. Three of them are easy, all right? Three of them are all made, actually four of them are made by an enzyme known as ribonucleotide reductase. But it turns out there's five. I'll tell you about that in a second. But four of the five are made by the enzyme known as ribonucleotide reductase. You can abbreviate that and call it RNR if you want to. RNR. Now, what does ribonucleotide reductase use as a substrate, and what does it produce? All right. Well, the starting material is a ribonucleoside diphosphate. That would include something like ADP, GDP, CDP, UDP. Okay? Notice I didn't say TDP because we don't make TDP in the synthetic pathways I've just shown you. All right? We make TDP separately. I'll tell you how that goes. So if we take, for example, ADP and we treat it with ribonucleotide reductase, we end up with deoxy-ADP, that is DADP. And the difference is that this ADP has an oxygen at position number 2. The DADP is lacking that oxygen. So that stands for deoxy. That means that that oxygen is gone. The DADP can be converted by NDPK into DATP. And DATP is necessary to make DNA. To make DNA, we have to have DATP, we have to have DGTP, we have to have DCTP, and we have to have DTTP. Well, I haven't told you how we make, yes? Say all those again, yeah. Would you like me to say them as fast as I possibly could? Okay. So, DATP, DGTP, DCTP, and DTTP. DTTP, yep. Okay? Now, I haven't told you how we make the thymine nucleotides yet, so that's what I'm going to tell you in a minute, all right? The thymine nucleotides are actually made from the uridine nucleotides, the U's. The U's are turned into T's, and I'll show you how that happens. Can I go ahead? Or should I repeat it again? I'm good, okay. All right, so we have to synthesize thymine if we want to make DNA. Well, it turns out we can do that, and we can do that with the synthesis of thymine occurs by a very interesting pathway. Okay, thymine comes from, not from DUDP, it comes from DUMP. Okay, dump. Yes, that's exactly what you're going to do with this information as soon as the exam is over, right? Just poof, it's gone. All right. DUMP, all right, is converted into DTMP, all right, by an enzyme known as thymidylate synthase. That's a simple name, thymidylate synthase. Let me spell that. If I spell it, then you'll have to know it. But if I don't spell it, you still might have to know it. Okay, we'll throw that one out. How's that? Okay. What's that? Are these legal Scrabble words? They are when I play Scrabble. I never lose at Scrabble. Because I make up words and people never know if I'm making them up or not, right? I have a student that I was very fond of, and she was one of my TAs, and she was a very good student. And she bragged that she had never been beaten at Scrabble before. And so I'm pretty good at Scrabble myself, and I told her, I said, well, I've never been beaten at Scrabble before either. Okay? So this meeting of the minds, we decided that one night we're going to play each other in Scrabble in this big face-off, right? And so we did this, and we played Scrabble, and I've never in my life had this happen. We got all the way to the very end, and we tied. I have never tied anybody in Scrabble in my life, and I tied her on that. So we decided after that happened that we would never, ever play each other again because there was no way of ever doing better than a tie, which is what we did. Pretty cool. All right. You guys don't want me to finish this, do you? All right. Let me show you one thing. Okay. This guy right here, this big mouthful, that's a folate. I told you need to have folates to make nucleotides. Folates have appeared other places in the pathway. I didn't show you those, but here's one place where you need a folate. Because what's this guy doing? Well, it's putting a methyl group right on there. The difference between a DTMP and a DUMP is a methyl group, and that's the methyl group right there. Now, I want to say one more thing about folates because it has very interesting and very important health considerations, and it's actually shown right here. Okay. Folates are critical for us to live. I've talked about that a little bit already. All right. And it turns out that folates, like other things, are recycled. All right? So here's a variety of different folates. A variety of different folates. So in the reaction that you just saw, all right, this guy here is getting converted into this guy over here. So what? Well, if I don't convert this guy here back into this, the cell will run out of this guy. And this guy, this tetrahydrofolate, is necessary for the synthesis of DTMP. Okay? You with me? In other words, if I stop this cycle, the cell will not be able to make nucleotides, and specifically it's not going to make DTMP, because everything's going to end up over here, or it's going to end up over here, but it's not going to make it back to here. Well, this turns out to be a very good strategy for chemotherapy. Very good strategy for chemotherapy. You have cancer, you have uncontrolled cell growth. What do cells need to grow and divide? They need to replicate their DNA. What cells are most susceptible to an inhibition in nucleotide synthesis? An aggressive cancer cell. Okay? So what we do in some types of chemotherapy is we treat them with an artificial folate to inhibit the enzyme. The artificial folate that we give them is called methotrexate. That you should know. M-E-T-H-O, trexate, T-R-E-X-A-T-E. Methotrexate inhibits this enzyme right here. This enzyme you can call DHFR. Well, you've just stopped the cycle. So what happens is if you have somebody who has an aggressive cancer that's not responding to other things, you can give them methotrexate for a short period of time. If you give it to them for a long period of time, it would kill them, because you have to have DNA and nucleotides for your own healthy cells. But for a short period of time, methotrexate will stop this enzyme and stop those cancer cells from being able to replicate their DNA. The cancer cells will die more rapidly than will normal cells. And when the cancer cells die, then hopefully you don't kill too many normal cells and everything's okay. Usually you do kill normal cells, and that's one of the reasons with some types of chemotherapy like this, people get sick, they lose their hair, etc. But the aim is preferentially killing cancer cells. So methotrexate is a very powerful drug, and methotrexate is also used for other purposes in very, very tiny amounts. So there are some autoimmune diseases, for example, that they use to treat methotrexate and things like that, but in much lower doses than they would use for chemotherapy. Yes? Jerry? I don't know a name. I'm sorry. Yeah. There's not a name given to it that I'm aware of. All right. Well, we started the term with a song, so I figured we would finish the term with a song. And I will encourage you to come up and get some cake as well. Let me get the music. This is a recording, actually. You can sing along. Anybody know the original song? It's an old song called They Call the Wind Mariah. It's not my most upbeat song. That's it. Same thing. Repeat. On that happy note. Okay, if you didn't get a note card, come and get a note card, and I will see you next week.